Hello, my name is Enzo Perez, and today I'm going to be talking about DNA structure and replication. DNA, or deoxyribonucleic acid, is a hereditary material that contains genetic information. A DNA nucleotide is composed of a nitrogenous base, a 5-carbon sugar, and a phosphate group. In DNA, the 5-carbon sugar is deoxyribose sugar. The nitrogenous bases include adenine, thymine, guanine, and cytosine. There are two forms of nitrogenous bases, purines and pyrimidines. Adenine and guanine fall under the purine category, while cytosine and thymine fall under the pyrimidine category. Purines have a single ring structure, while pyrimidines have a double ring structure. A pyrimidine cannot pair with a pyrimidine, and a purine cannot pair with a purine. A pyrimidine must pair with a purine. DNA has a double helix structure, which is antiparallel. Meaning that the strands run in opposite directions. The 5' end pairing with the 3' end. With the two strands antiparallel to each other, the nitrogenous bases of the two strands can bond to each other. Adenine always pairs with thymine, and cytosine always pairs with guanine. The nitrogenous bases are paired together with hydrogen bonds, adenine and thymine having 2 hydrogen bonds, and cytosine and guanine having 3 hydrogen bonds. Phosphodiester bonds are also present within the DNA structure, which are bonds that are present between DNA nucleotides, forming the sugar-phosphate backbone. Chargaff's rules are present when it comes to DNA, stating that the base composition varies between species and within a species. The number of adenine and thymine bases are equal, and the number of cytosine and guanine bases are also equal. Watson and Crick developed a model that was able to overall display Chargaff's ratios and explain them. DNA partakes in an event called DNA replication, in which genetic information is duplicated in order for an organism to pass down its genetic information. DNA replication is essential for biological inheritance. It takes place in the nucleus of eukaryotes and the cytoplasm of prokaryotes during interphase, specifically the S phase of the cell cycle. DNA replication occurs in the S phase. DNA is replicated in a semi-conservative fashion, meaning that after the 2 copies of the original DNA molecule are produced, each molecule contains a strand from the original DNA molecule. Each strand in the DNA double helix serves as a template in order to form a complementary strand, leading to one old and one new strand in the newly produced double helix. The directionality of the DNA molecule is able to severely affect and influence the replication process. DNA polymerase is an important enzyme to process of replication. It adds complementary nitrogenous bases to the template of the growing DNA chain. However, DNA polymerases can only add nucleotides to the 3' end of the primer or a growing DNA strand. As a result, a new DNA strand can only elongate in the 5 to 3 direction. On one strand, DNA polymerase 3 is able to stay in the replication fork and on the template strand, continuously adding nucleotides, which is called the leading strand. However, in opposition, the other strand must also be elongated in a 5' and 3' fashion, which means that the DNA polymerase must work along the other template strand away from the replication fork, which is called the lagging strand. The lagging strand is synthesized discontinuously in a series of segments, called Okazaki fragments, in opposition to the leading strand that is synthesized continuously. Within eukaryotes, Okazaki fragments are approximately 100 to 200 nucleotides in length, while in prokaryotes they are approximately 1,000 to 2,000 nucleotides in length. There are a multitude of enzymes involved in the process of DNA replication, some including DNA helicase, topoisomerase, single-strand binding proteins, primase, DNA polymerase III, DNA polymerase I, and ligase. DNA helicase is the first replication enzyme that acts on the ORI, or origin of replication. It functions as the enzyme that unwinds the DNA, breaking the hydrogen bonds, separating the parental strands, and allowing them to be used as template strands. In order to keep the strands from pairing with each other after the unwinding, single-strand binding proteins come into play, binding to the unpaired DNA strands in order to keep them from repairing. With the unwinding of the DNA molecule, tighter twisting and straining may occur ahead of the replication fork. In order to prevent the straining, topoisomerase relieves the strain by breaking, swiveling, and rejoining DNA strands. The enzyme primase synthesizes RNA primer to the template strand, which in turn provides DNA polymerase III with a template to use. However, RNA primer is added to the lagging strand in a series of Okazaki fragments. DNA polymerase III is the enzyme that adds DNA nucleotides to the RNA primer from the 3' end. Additionally, DNA polymerase III is able to remove incorrect nucleotides that are accidentally added to the chain. DNA polymerase III adds nucleotides to the lagging strand in a series of Okazaki fragments, slowing the process. After DNA polymerase III forms an Okazaki fragment, the DNA polymerase I proceeds to replace the RNA primer of the adjacent primer with DNA nucleotides. Because DNA polymerase I cannot join the Okazaki fragments, ligase comes into play, joining the sugar phosphate backbones of the Okazaki fragments into a continuous DNA strand. DNA replication is a fascinating yet complex process that has a plethora of components to it. Each enzyme must work harmoniously with one another in order to achieve the final result, which is to produce enough DNA in order to pass on genetic information to the next generation.